Okay, guys, so we're going to start DNA replication today, but this is going to be a multiple lesson topic. We're going to kind of start of how DNA replication initially starts and kind of how it happens on the big picture today. And then next week we'll talk about the rest of replication. Okay, so this is not going to be the entire process. So the first thing we're going to talk about is the idea that DNA is semi-conservative. Okay, and what I mean by that is the replication is semi-conservative. Okay, so I like you to kind of follow along with my diagram here, and it'd probably be a good idea to copy out the diagram as well on your own. Okay, because we learn a lot better that way. So the first thing I'm going to do is I'm going to say, okay, what parental strands are going to be in red, and daughter strands are going to be in blue. Okay, so let's draw our original piece of DNA here. Okay, so I've got my original piece of DNA here. And I'm just going to put some letters in here. So I'll put A, G, C, C, A, T. Okay, and then corresponding to it, I know that if that's an A, that must be a T. If that's a G, that must be a C. That must be a G. That must be a G. That must be a T. And that must be an A. Because they have to match up, right? That's how DNA is made. Okay. And we also know that there is 2 bonds between these, 3 bonds between each of these, connected together. Okay. So there's my original DNA strand. Now what we're going to do is we're going to duplicate it. Okay, we're going to replicate it. Now this is how replication works. Okay. What's going to happen is the first thing that's going to happen is we have to break these bonds. Okay, so we're going to go in and we are going to break all of these bonds. And what that's going to cause is it's going to cause the 2 strands to separate. Okay, and so now we're going to redraw our strands here. I'll separate it. So this is going to go, and now I'm going to have my strand up here, which is A, G, C, C, A, and T. And again, this was my original strand, okay, or my parental strand, okay, that's why I have it colored in red. And then my bottom piece here, it's going to be T, C, G, G, T, A. Okay. So what has happened is we basically opened up the DNA. Okay. Now we're going to start replicating it. Okay. So when we say that it's semi-conservative, what that means is that we are going to conserve at least part of the original strand. Okay. So this one right here that I'm pointing to, this is part of the original, and that's part of the original. We call those parental strands, okay? And they're the original DNA helix. That was one part of the helix, that was the other part of the helix, okay? Now, when replication happens, okay, and we eat a whole bunch of bases, so we eat a whole bunch of A, G's, C's, and T's, and we eat like our bananas, and we take their A, G's, C's, and T's. We eat a piece of chicken, we break down, and we have all their bases. So now when we want to replicate our DNA, we just basically go to our stockpile and we say, okay, I'm going to replicate the DNA here. And all I'm going to do is I'm going to match up T, C, G, G, T, A. And this is one double helix. So this will go in cell 1. And then this one down here also replicates. It just grabs its stockpile. We've got A, G, C, C, A, T. And now we have 2 pieces of DNA. So we've now replicated. This was our original double helix right here. Our original double helix, and it was bonded, and then we opened it up. And we've replicated the DNA, and now we have 2 double helixes. And they're exactly the same. Look at the coding. It's exactly the same. Okay? Now, when we say it's semi-conservative, why we're saying it's semi-conservative is because you have the new pieces of DNA, the new double helixes, are half original, right? This is our original part right here. So it's half original, and then half brand new. This is the brand new part, right? The bases that we just ate and we broke down. Okay? And that's how DNA essentially happens, is you open it up, and then you basically make a brand new line of DNA on both sides. Okay? And then this would go into, again, the other cell. Okay? So you had one cell with the DNA inside it, you doubled your DNA, and now you can have two cells, each with a copy of its own. Okay. Now, let's go through replication. So replication happens in 3 phases. Okay. Today's lecture is only going to go through 1, and the only phase that we're going to go through today is the initiation phase. Oops, wrong one. It was on the highlighter. This one right here. We're going to go through the initiation phase today. So this is like preparing the DNA to duplicate. Okay. So let's go through it. It's not very complicated. All right, so initiation phase. So replication cannot begin anywhere on the double helix. You can't just start replicating the DNA anywhere, okay? But only in certain spots, which we call replication origins. Now, what these replication origins are, there's basically just a whole bunch of AT bonds, okay? When you look at the AT bonds, so the ones that I'm highlighting down here, there's only 2 connections compared to a GC connection where there's 3. So it's a lot easier to break open the DNA when there's only 2 connections. So where DNA opens up originally and starts to replicate is always where there's a whole bunch of A's and T's, and we call that a TATA box. Okay, and the TATA box is just TATTA. It doesn't have to be in that order. We just call it a TATA box, where there's a whole bunch of A's and T's all in a row, meaning there's a whole bunch of loose bonds and they can break apart. Okay, so that's where DNA starts to replicate. So you have to kind of find a TATA box, a whole bunch of T's and A's, and that's where you can, the location that you can open up the DNA. Okay, so now we found that location. We found the TATA box. Next step, go back. It's the part that's going to go through and break all of these. So there's an enzyme that's going to come in and just basically cut all of these things, all of these hydrogen bonds. And what that enzyme is once replication origin has been found, once you found that TATA box, 3 important proteins get involved. Okay, so it's going to be 3 proteins that we're going to go through today: helicase, topoisomerase, and single binding proteins. Okay, so the first thing that we're going to go through is helicase. So helicase is pretty simple. Okay, it is an enzyme that basically breaks the hydrogen bonds between the bases. Okay, so it is the part that's going to go through the DNA. Okay, helicase. That's helicase's role. So that's its role right here. It's going through and it's cutting those hydrogen bonds. So right up here, I would have a series of, bases. And then I'd also have the corresponding ones here. Okay, that's what helicase. And this is helicase again right here. It's just basically, this is the part that cut all the hydrogen bonds that were there open. Okay. All right, so we cut them open. Helicase does. The second molecule, DNA gyrase, also known as topoisomerase 2, all it does is a little bit downstream. It's in front of helicase. Before helicase even opens up the DNA strand, we have to make sure that the DNA doesn't accidentally just start tearing prematurely. Okay, because as you open up the double helix, there's going to be pressure, and you don't want the DNA to tear. You actually want to make sure helicase cuts it as opposed to it accidentally just ripping. So what the only role of topoisomerase 2, or DNA gyrase, is to relieve the tension. Okay, so it just goes right in front of helicase, and it just kind of makes sure that it doesn't tear. It relieves the tension in front of helicase. Okay, so that's 2. Now we only have 1 thing left. The single binding proteins. So single-stranded binding proteins. So these are what these things are right down here. And they also have a very simple role. All they do is prevent these 2 strands from reforming. Okay? Once helicase has gone past them and cut them open, they want to make sure that these things don't start reforming before you have a chance to replicate. So just to prevent the double helix from reforming and from keeping them actually apart, we use single-stranded binding proteins. So these prevent the single strands of the DNA helix from reforming the hydrogen bonds between the bases that helix cut and becoming double helical again. Okay? And that's all we have for initiation. Okay, that's all we really want to go over for initiation. Was those 3 parts. So we're going to kind of look at, this is a good picture because it kind of shows at least the 3 proteins that we looked at. First is topoisomerase. So this is during initiation. And we have this little ring here that's basically relieving the tension ahead. Okay. Then we've got helicase. So let me just highlight this. We've got helicase here. We got helicase, topoisomerase. And helicase, again, it's just going through and cutting. Okay, it's cutting all the hydrogen bonds, opening up that double helix. You can see down in this area, this is actually where the rest of the replication is going to happen. We're not there yet. Helicase is cutting. And then the last thing that we wanted to look at was single-stranded binding proteins, and those are right there. And that's just preventing the DNA from reforming before it gets a chance to replicate, which is happening down here. Okay. And that's the only parts of the initiation phase. Again, it's not very complicated. Once we get into the elongation phase, which is going to be next, that's where it gets a little bit trickier. But again, we're going to be kind of keeping this a higher level than what we would normally be in class due to the circumstances that we have. Okay, thanks guys. Let me know if you have any questions.